Assalamualaikum warahmatullahi wabarakatuh. We are from AS20158 and today we are presenting the gene cloning and DNA analysis in agriculture. My name is Nur Rabatul Misha binti Jamaluddin.
My name is Nur Afifah binti Syahrul Wanri.
My name is Nur Syafiqah Hadira binti Muhammad Syazali.
And my name is Nur Shakira Atikah binti Muhammad Rozi.
So, first, let me introduce you to gene cloning. What is gene cloning? Gene cloning is also known as molecular cloning. It refers to the process of isolating a DNA sequence of interest for the purpose of making multiple copies of it. The identical copies are clones. In 1973, Stanley Cohen, which is in the left side of the picture, and Herbert Boyer, on the right side of the picture, developed techniques to make recombinant DNA, which is a form of artificial DNA. Under the gene cloning, we have two strategies or techniques, which are gene addition and gene subtraction. Gene addition is the cloning strategies to alter plant characteristics by providing more new genes into the cell, while the gene subtraction is the inactivation of the existing genes. Before we go through in detail with the techniques of the gene cloning, let me explain about the basic mechanism of gene cloning. There are a few steps of gene cloning, which is the first, choice of host organism and cloning vector. The host organism and cloning vector will be chosen prior to gene cloning. For example, in this diagram, the host organism is the E. coli cell, while the cloning vector is the bacterial plasmid. Next is the preparation of vector DNA and preparation of the desired DNA. The vector DNA usually used is the plasmid taken from the bacteria that contains the ampicillin and the LAC-Z gene, while the desired DNA is the DNA containing the gene of interest. Both of the components, which is the plasmid and the gene of interest, is cut with the same restriction enzyme, that leaving them with the sticky ends. Next is the creation of recombinant DNA with DNA ligase. Both of the DNA components will be mixed and joined by base pairing. Then, DNA ligase will be added to bind them together covalently, forming the recombinant DNA. The gene of interest fragment will be bind into the plasmid vector with the non-functional LAC-Z gene. Next, the plasmid recombinant will be introduced into the host organism, which is the E. coli cell in this case. The plasmid recombinant will be put into the LAC-Z bacteria by transformation. And next, selection of organisms containing vector sequence. The E. coli cell will be plated onto the medium with ampicillin and X-gal, which acted as the indicator. The X-gal will determine the positive results of the cloning, which will resulting in the white colony. The clone will be identified that the cells containing the recombinant plasmid by their ability to grow in the presence of the ampicillin and their white color. And lastly, the screening for clones with desired DNA inserts and biological properties. The identified clone will carry the gene of interest, and the bacterial clone will carry many copies of the human genes of interest. So next, I will pass to Ms. Atika to explain more about the techniques of the gene cloning.
We will now talk about one of the techniques of gene cloning, which is the gene addition. As mentioned by Ms. Misha before, it is a cloning technique to introduce one or more new genes into a plant that coded for a useful characteristics that the plant lacks. As example, developing plants that are resistant towards insects and production of herbicide-resistant crops. The major method used for inserting genes into the plant genome is by using the plasmids of bacterium, Agrobacterium tumefaciens. So why? Why do we use Agrobacterium tumefaciens? It is a soil-dwelling bacterium that invaded plant cells and the cause behind tumor induction. Its plasmids carry the genes for tumor induction and inserted itself into the plant chromosome. Thus, the plasmid is called as TI plasmid. Now, what are the advantages of using the Agrobacterium tumefaciens mediated transformation as compared with direct gene transfer? First, it is integration of low copy number of tDNA into plant genome, stable gene expression, and transformation of large size DNA segments. Now, this is the use of TI plasmid. As we can see on the upper diagram, the Agrobacterium tumefaciens is the cause for the disease crown gall in plants. We will now move on to the mechanism of gene addition. How do they work? Firstly, the TI plasmid will be removed from the Agrobacterium tumefaciens bacterium, and the tDNA and foreign DNA, being our gene of interest, will be cut by the same restriction enzyme. The foreign DNA will then be inserted into the tDNA of the plasmid and ligated using DNA ligase. The TI plasmid carrying the foreign gene will then be introduced in the chromosome of plant cell and grown in a culture. Then, regeneration of plant occurred and will result into a plant that carry the new clone genes and may express it as a new trait. As example, they are resistant against herbicide and able to produce their own insecticide. This diagram shows the basic making of a transgenic plant. Firstly, designing and making the gene construct. Second, introduce the transgene to plants by using Agrobacterium tumefaciens that can be found in the wild or by using an alternative method called biolistic method. Thirdly, grow the transgenic plantlets. Fourth, confirm the plants are transgenic. And the last step would be we can conclude what the transgenics can tell us. I will now pass the presentation to Ms. Shafiqah.
Okay, I will explain on gene subtraction. Gene subtraction is an inactivation of target gene. There is no removal of gene in gene subtraction. Antisense is one of the technique under the gene subtraction. How this technique works is by gene interest that will be cloned is ligated into vector in reverse orientation. The clone gene is when the clone gene is transcribed, the RNA synthesized is the reverse complement of the mRNA from the normal sequence. The antisense of RNA can cause hybridization between antisense and sense copies and block the translation process and the production of the protein. Okay, as we can see here, we can see the normal gene and antisense gene in reverse orientation. In normal gene, mRNA will be produced. In antisense gene in reverse orientation, the antisense RNA will be produced. When these two RNA present in one cell, in the same cell, they will form a duplex formation. When this mRNA and antisense RNA form the duplex formation, they will cause the prevent ribosome from binding. Double-stranded RNA can be transported to cytoplasm, and mRNA will be degraded by RNase-H. When this event occurs, there will be no protein production. Next, we will see antisense technology application, which is flavor saver tomato. The aim production of flavor saver tomato is to improve the shelf life of the tomatoes. The process of fruit ripening is adding by enzyme polygalacturonase, or we know as PG. This enzyme will degrade the cell wall of the fruit, also known as pectin, and will causes the softening process. The mechanism of flavor saver tomato production is P-TOM6 genes, which is responsible for PG enzyme, will be isolated from the tomato and will be cloned. The PG gene will be transferred to a vector bacteria and undergo reverse transcriptase to produce complementary DNA, or we know as cDNA. The cDNA will be introduced into a fresh tomato plant, and in the plant, the cDNA of PG encodes for antisense mRNA, which is complementary to sense mRNA. The hybridization between sense and antisense mRNA make the sense mRNA ineffective and prevent the translation of mRNA for PG production. Thus, when there is no PG production, pectin will not be degraded. The process of ripening can be delayed. Next, we will see the flavor tomatoes visualization. This here we can see the normal tomato and the transgenic tomato. I will pass the presentation to Shakira.
So I will present the benefits of the gene additions and the gene subtractions. So the first one is the benefits of the gene additions. So the gene addition will make the plants resistant to insects. So the genes that we want is the genes that encode for the delta endotoxins and proteinase inhibitors. This is the example of the gene we want it to be encoded for. And the source we get it from the Bacillus thuringiensis and various of legumes. So for example is the corn, where the Bacillus thuringiensis corn, which is the corn GMO, is a corn that produces a proteins that toxic to a certain insect species, but not to human, pets, livestock, or other animals. So these are the same types of proteins that organic farmers use to control the insects' pests, and they do not harm other beneficial insects such as ladybugs. So the GMO corn reduce the need of spraying the insecticides while still preventing insect damage. So the next benefits is the plants will be resistant to fungi, and the example of the genes encoded for is chitinase. So which is we get it from the tobacco, and the example is peanuts, because based on my finding there's a study done where the peanuts, fungal resistant peanuts, was developed by inserting the tobacco chitinase gene. The next benefit the gene addition is for the sweetness, where the genes we want to encode for is thaumatin and monoline. So the thaumatin, we get it from a Tomatococcus dainelli, which is a type of plant. So thaumatin is a low-calorie sweetener and flavor modifier. So based on my research, I found that a paper that done a study where they insert the gene that responsible for the production of thaumatin into the tomatoes, and as the result, all the panelists that actually taste the tomatoes describe it as sweeter than the control tomatoes. The next one is gene addition will make the plants resistant to the virus. So the genes we want it encode for is virus called protein, and we get it from the virus of viruses. So the example is plum and papaya. As we know that papaya is actually, this happened in Hawaii, where papaya's plants grown in Hawaii were resistant to attack of some viruses, which is all started in 1990s, where the ring spot virus disease nearly wiped the Hawaiian papaya crop. So what they do is they make a GMO papaya named a rainbow papaya, and this papaya is resistant towards the ring spot viruses, and it eventually saved the papaya industries in the Hawaiian island. So the last one, which is the last benefit that I will show about the gene additions, is plants will be resistant to the herbicides. So the example is soybean, where we want the genes for the 5-enopyruvyl shikimic acid 3-phosphate synthase, which it will make the soybean become the glyphosate herbicide tolerance. And this we get it from the bacterium. The next is the benefits of the gene subtractions. So the first one is the gene subtractions will prevent the discoloration in fruits and vegetables, and the target genes is the polyphenol oxidase. So the next one is it will delay a fruit spoilage, which it target the polygalacturonase, which delay the fruit spoilage in the tomatoes. The next one is the gene subtraction will reduce the starch content in the vegetables, which it target the starch synthase genes. And the final benefits, or the last benefits that I will share about the gene subtractions is it will modify the flower's colors where it target the gene chalcones, which is the genes that are responsible, okay, for the various decorative flowers.
So I guess that is all from us. Thank you for listening, and we hope all of you will understand how gene cloning works in the agriculture sector more now. Thank you.